Second mini lecture, we're going to delve a little bit deeper into the innate immune response. We're going to try to understand better how complement induces its functions. What are some of the key ways in which the innate immune system recognizes pathogens? The role of cytokines and chemokines in inducing an inflammatory response, and the effector functions of the innate immune response that result from that. Pathogens can be recognized immediately by the innate immune system, and we're going to focus on two components of that recognition: recognition by complement and recognition by pattern recognition receptors. So let's go back to our compromised epithelial barrier with an infection with some bacteria. These bacteria can be recognized immediately by complement components. They can also be recognized by innate immune cells bearing a class of receptors that we will term pattern recognition receptors. So let's start thinking about complement first. So as a review, complement is our inactive proteases that are found constitutively in plasma and interstitial fluid. They can be converted to active proteases by recognition of pathogen surfaces. And activated complement components induce three things. One, they induce lysis of pathogens directly. Second, they induce the phagocytosis of pathogens by innate immune cells. And third, they increase inflammation by inducing vasodilation of local capillaries. So let's look at those functions in a little bit more detail. So here we have a bacteria, and there are complement components that can be activated and undergo autocleavage when they recognize that bacteria. And so here we have, for example, some complement components that have generated cleavage components as part of a proteolytic cascade. Some of those cleavage products can attach covalently to the surface of bacteria. And once they do that, they can act as a class of molecules that we call opsonins, or in other words, molecules that enhance the ability of phagocytes to recognize this cell. So here's a neutrophil, for example, that expresses complement receptors. These complement receptors can recognize these complement fragments bound to the surface of this cell and enhance the ability of this neutrophil to phagocytose this bacteria. Neutrophils and macrophages are really good at this. Another consequence of this, and I guess I should mention, here's just a picture now of this neutrophil that has taken up these bacteria into the phagolysosome. Another consequence of coating this bacteria with these complement fragments is that it in turn recruits more complement components that lead to the lysis of this bacteria. And then the last part of this is that some of these fragments, such as these initial fragments up here, can directly induce inflammation because they act on local capillaries and induce their vasodilation. So important to note, complement is induced by direct recognition of pathogen surfaces. Complement is also induced, activation is induced by recognition of pathogens that are bound by antibodies. And we'll talk a little bit about that when we get to the adaptive immune response. Complement also mediates opsonization. So complement fragments that coat bacteria are called opsonins. Opsonins are recognized by receptors on phagocytes, and they enhance phagocytosis. So the process of coating a pathogen with these molecules is called opsonization. Some types of antibodies are also opsonins. Opsonins are basically molecules that enhance recognition by phagocytes. Okay. So let's now turn our attention to pattern recognition receptors. Here we have a macrophage that can directly respond to these bacteria. How is that possible? Well, bacteria and viruses have a number of unique structures that can be recognized by the immune system. And this is just a brief introduction to some of the things that you will learn more about in host and defense. But these structures are often referred to as pathogen-associated molecular patterns, things that are expressed by the pathogens, but not by commensal organisms and not by mammalian cells. This includes molecules such as lipopolysaccharides and other lipomolecules, sugars that comprise the bacterial cell wall, such as peptidoglycan, proteins that form the structure of this tail called flagellin, fungal sugars, unmethylated CpG DNA structures, and in the case of viruses, double-stranded RNAs or single-stranded RNAs that are uncapped, and other strange nucleic acid structures that can occur during viral replication in mammalian cells. And our cells express a variety of these pattern recognition receptors, each specific for one of these broad molecular patterns. For example, pattern recognition receptors that might be specific for lipopolysaccharides, or specific for double-stranded RNA, or specific for bacterial nucleic acids. And each of these pattern recognition receptors can be found in different locations. Some are expressed on the cell surface, some are expressed in the cytosol, some are expressed in endosomes. When these pattern recognition receptors are triggered, they can induce an inflammatory response. It's important to note that all of these pattern recognition receptors are encoded in the germline DNA, meaning that we can't have an infinite number of these receptors. And so the strategy of the innate immune system is to have these receptors that are broadly specific. So an innate receptor that's specific for lipopolysaccharide is not going to distinguish the type of bacteria that's making that lipopolysaccharide. It's going to be able to be triggered by any bacteria that makes lipopolysaccharide. But when it is triggered, it can induce cytokine and chemokine responses. And so here's a macrophage that recognizes these bacteria and starts to make these inflammatory cytokines. These inflammatory cytokines and chemokines can induce activation of endothelial cells and vasodilation of capillaries and inflammation. And so here's a picture of how that might happen. Complement fragments and inflammatory cytokines can act on local capillaries to induce the activation of this endothelium and the vasodilation of these vesicles. And the result of that is that it makes it a lot easier for circulating innate immune cells, such as neutrophils, to slow down, attach to these endothelial cells, squeeze in between these tight junctions, and migrate into this tissue. Once they do that, they can follow along this chemokine gradient to the site of pathogen replication. And this is essentially how an inflammatory response is induced. And once these neutrophils there and these macrophages are there, then they can mediate their important effector functions that eliminate this pathogen. It's important to note that other leukocytes, lymphocytes, monocytes, eosinophils, they all use chemokines to traffic from one place to another. The nature of the chemokines may differ for different cell types, but the basic mechanisms are quite similar. So this is a review from the Intro to Immunology mini video that reviews some of the key effector functions of innate immune cells once they traffic to this site of pathogen replication. That includes phagocytosis, degranulation, spitting out extracellular DNA traps, or direct cytotoxicity in the case of viral infections. And all of these cells migrating into this site of pathogen replication leads to inflammation. And inflammation has certain key hallmarks, and this includes rubor, or redness, which occurs because of vasodilation that leads to increased blood perfusion. Also tumor, or swelling, which occurs because of increased plasma leakage into the tissue, again due to vasodilation and leukocyte recruitment. Heat, or calor, which results from increased vasodilation, perfusion, and metabolic activity due to leukocyte infiltrate. And 4, dolor, or pain, which can result from inflammatory cytokines that bind to nociceptors, leading to localized pain. And this results in a local inflammatory response. So this leads to activation of local immune cells. It enhances their activity. Activation of the vasodilation of endothelial cells enhances trafficking of leukocytes from the blood into the tissue. These leukocytes then can arrive to the site of pathogen replication through chemotaxis by following along that chemokine gradient. And once at that site, they can mediate destruction of the pathogen. So you can think of macrophages, which are tissue resident, as the early warning system, and neutrophils, which are in circulation, as the first responders. So it requires macrophage activation to pull in neutrophils to that site and generate an inflammatory response. Now, inflammation can have systemic effects as well, especially if it is prolonged or if the infection is unresolved. Some of those systemic effects can lead to inflammatory cytokine availability systemically and in circulation. One of the things that can happen is this can lead to release of neutrophils from bone marrow, which leads to an increased number of neutrophils in circulation. Sometimes inflammatory cytokines can induce increased hematopoiesis, and if this is a prolonged response, can lead to increased numbers of immature neutrophils or bands in circulation because the hematopoietic system can't keep up with the demand. Systemic effects of inflammation also include fever and rashes due to vasodilation. And lastly, harmful systemic effects such as sepsis and septic shock can occur in severe cases. Okay, so that is the end of our intro to the innate immune response. Our next mini lecture will delve into the development of the adaptive response.